Okay. Hi, everyone. Initially, I would like to thank the organizing committee for this inspiring and collaborative platform. It's a pleasure to share my work titled "Did You Read the Room? Really, Sediment, Geochemical, and Phytolith Evidence from a Neolithic Floor Context." Oops. First, I would like to provide an outline for my talk. I will begin with the brief introduction, the significance of spatial analysis, and main research questions of my research. Afterward, I will present the combined methodological framework. Next, I will introduce to Aşıklayık, specifically building 45 from 8 millennium BC settlement. Followed by the results, integration of them, and the key findings. And I'm aiming to conclude with a reflection on entangled life phase in Neolithic floor context. Can we truly read activity areas from floor deposits? In archaeological context, interpretation based on single proxy approaches often remain limited as they risk oversimplifying complex depositional process and use life processes as well. This study therefore aims to integrate multiple proxies to refine the macro-contextual interpretation of a Neolithic room. I argue that floor deposits should not be understood as a passive accumulations, but rather as chemically and botanically structured archives that record traces of everyday practice. To investigate macro-scale activity areas, sediment samples were collected using a grid-based sampling strategy to capture spatial variability across the floor. Elemental concentrations were measured with a highly sensitive instrument and visualized by interpolating elemental mapping in GIS. Target elements are analytically significant for identifying anthropogenic inputs, such as organic waste, storage, burning residues, food preparation, or dung accumulation. On the other hand, phytolith analysis was conducted using standard heavy liquid separation with floor sediment subsampled. Sorry, macrobotanical data helped to detect activities related crop processing, storage, floor matting, and the presence of plant-based artifacts. As I mentioned before, my case study is Aşıklayık, which is located in central Anatolia in modern Turkey or Turkey today. Aşık is a well-studied preparatory Neolithic site that experienced long-term continuous occupation, forming a coherent cultural sequence without any major interruptions. The focus of this study is level 2. You can see here. Above ground quadrangular buildings characterize a dense settlement pattern. This settlement layout is also known as Central Anatolian model, in which building entrances are from the roof level. These flat roofs also served as a courtyard in the feasible weather conditions. Today, I'm focusing on phase D, during which storage units emerge as an architectural unit within rooms, though they were rare. This corresponds approximately to 7,500 BCE. Building 45, a double room building with integrated storage bins at the northeast and southeast corners in the east room. The west room was exposed and documented in the old system. The western part of the room is damaged, therefore it was not included in the analysis. 39 sediment samples were analyzed to understand the geochemical composition. Accordingly, the presence of embedded chemical residues suggests that the southwest corner of the room may have been used as a food preparation area. However, geochemical analysis revealed no other clear spatial pattern in the room. For this reason, I extracted phytoliths from floor sediments, first and foremost to gain insight into taphonomic effects on the plant assemblages. I tested the correlation between identified morphotype counts and phytolith concentration. The correlation index indicates a weak negative correlation. So, in general terms, the room's plant assemblage is dominated by Poidiae, which include cereals such as wheat and barley, and are followed by the Panicoideae subfamily. Related to cereal remains from the room's floor and storage surfaces, I want to show some well-preserved silica skeletons. Luckily, the environment of Aşıklı Höyük enhanced the robust silicification of phytoliths. Here you can see an elongated dendritic cell belonging to wheat. Another example of wheat silica skeletons can be seen here, together with another conjoined cells. Now I would like to show you the spatial distributions of plant macro remains. The distribution, JS distribution map demonstrates phytolith concentration on floors. Blue indicate high values. Based on these distributions, phytolith concentration is in the southern part of the room, particularly at the threshold. It is important to highlight the presence of a grandstone at the threshold. Here, on the bottom, Panicoideae grasses are distributed across the floor. There's a peak at the northwest corner, and on the right, the Poideae subfamily, which is dominating the plant assemblage by being almost 45% of it. The distribution shows peaks several locations in the main area, including each storage bin. In terms of plant parts, the distribution grass, culms, and leaves in the room reveals an almost homogeneous pattern when we exclude the northeastern silo samples, suggesting similar use of the southeast silo and the main area. So you can see distribution of sedges and palms on these maps. Interestingly, sedges only occurred in the northeastern silo. Data indicate that northeastern silo was primarily used for cereal storage, dominated by barley, with some wheat, suggesting mixed or repeated storage episodes. The presence of cereal byproducts and sedges phytoliths points to organic containers or coverings, while sample variability indicates multiple use phases. Sporadic wild millets likely reflect weedy harvest inclusions or plant-based artifacts again, and palm phytoliths, which is non-local to Central Anatolia, indicate deliberate use of imported plant remains, materials, I mean. Overall, the room shows a well-organized spatial use with high cereal and husk phytolith concentration, indicating plant processing and storage. Such phytoliths restrict to the northeastern bin, suggest protective plant-based items, utensils, palm leaves, Phragmites, panicoid, and cereal stems were likely used for raw materials for basketry items. According to integrated data, distinct activity areas are evident. The southwestern corner served as a food preparation zone with an actively used threshold that reflects repeated plant circulation and grain stipulage or its product. The northwestern corner was a high traffic access point characterized by chemical depletion for certain elements and limited use due to the leather placement there. And palm phytoliths further indicate us to use the use of the non-local plant materials, pointing to long-distance perishable resource networks. Building 45 highlights human-plant entanglement at Aşıklı Höyük. Expanding cultivation led to construction of permanent storage units, which in turn created ongoing cycles of labor, maintenance, and cleaning that structure daily routines. The conversion of the southeast silo into the main processing area reflects continuous negotiation between human needs and material constraints. This study shows that combining geochemistry and phytolith analysis enables interpretation beyond the macro-scale evidence. Life at Ashıklayık was shaped by evolving relation among humans, materials, and domesticates, evidence for flexible storage systems, and the presence of exotic plant remains also suggest adaptive practice and potential Neolithic exchange networks. All in all, integrating dual proxies reveals Neolithic floors as records of everyday entangled lifeways rather than simple surfaces. I would like to thank you all finding bodies, and particularly I want to thank all the women who have touched my life, and thank you for your listening and your patience.